In this video, we'll show you a movie taken under the scope of the Meristem Excision technique for virus elimination treatment in grapevines as we use it here at Foundation Plant Services, UC Davis. My name is Sue Sim. A microshoot tip, which we also refer to as a Meristem tip, consists of the Meristem dome plus 2 to 4 pairs of leaf primordia. It measures less than half a millimeter. Here is a light micrograph of a tip. The virus is labeled with a fluorescent purple dye. In this case, the virus is a phloem-limited virus, so it's only infecting the protophlom cells. The really great thing about this photo is that it clearly shows that if you excise the Meristem dome plus a few leaf primordia at the top and grow it into a whole new plant, you've escaped the virus infection and have a clean mother plant to propagate from. For other viruses that are not restricted to the phloem, we hope that the cells in the tip are growing faster than the virus can infect them. Basically, it's propagation on a very small scale. The plants must be virus tested after treatment to ensure that the treatment is successful. So we start by collecting the tips that are about 2 centimeters long. It's important to keep them turgid, so we collect in the morning into a box with a wet paper towel in it. Then we bring them into the lab and work in the hood. From this point on, everything is done aseptically with sterile instruments. Next is the movie part. We start by trimming away the sides of the leaves near the tip. This saves a lot of time and makes it easier to remove the leaves. But Slava is very practiced at this. She's literally excised thousands of tips and has a very good idea of where the Meristem dome is located. She's trimming away the scaly leaves here, and she'll trim them off one by one until she gets closer to the Meristem dome. Over the years, she has developed really good techniques to increase efficiency and accuracy, and she can do anywhere from 70 to 100 in a day. It is very hard to make such precise cuts on such a small scale and takes a lot of practice. It's important to use a new sterile blade for each cut. We didn't do that in this video to save time, but for the final cut, it's especially important to avoid transferring any virus from older to younger tissue on the scalpel blade. She has a few more leaves to peel off, and she just increased the magnification from about 30 to about 60 or 70x. She's adjusting the orientation, and the last one, there's the dome. It's shiny and vibrant, surrounded by 2 pairs of leaf primordia. Including a few primordia increases survival, and she's going to cut it off soon and very quickly. The tip dries quickly in the air, so you want to place it on top of the medium soon after you excise it, and not too deep into the medium or it will drown. At this stage, it's barely visible to the naked eye. We incubate the tubes in a growth chamber and transfer the tips to fresh medium every 2 to 3 weeks. In 5 to 7 months, it develops into a little plantlet with roots and leaves. We then transfer it to potting mix and acclimatize it in a humid box. In this case, it's a upside down file box. And then in about 1 month, it grows into a 4-inch potted plant, and then it will be transferred to a gallon pot to grow larger and be subjected to a panel of tests. Some grape cultivars are vigorous and easy to excise and grow in tissue culture, such as Cabernet Sauvignon, and others are really very difficult. Every single vine you see here in the Russell Ranch vineyard has been through this therapy, so this technique has proved to be very successful and is used routinely at FPS. For more information, see our website at fps.ucdavis.edu.